Good morning, students. I'm Shomajit sir. Today I'm here to deliver my lecture on topic Search for Genetic Material. This is from the chapter Molecular Basis of Inheritance. So watch the entire lecture to get basics out of it. So without any delay, let's start with the topic Search for Genetic Material. So let's start. Let's start with the topic Search for Genetic Material. Here we'll see the experiments to prove which is the genetic material. Okay. So this experiment is also called Griffith's transforming principle, where Frederick Griffith in 1920 used mice and Streptococcus pneumonia. Okay, so Streptococcus pneumonia is a bacteria. So Streptococcus pneumonia has two strains. One is the smooth strain that are virulent in nature, and rough strain that are non-virulent in nature. So smooth strain virulent, it has a polysaccharide mucus coat, okay, and it causes pneumonia. Whereas the rough strain is non-virulent, it has no mucus coat and do not cause pneumonia. So you can see the mice and the mucus coat. You can see the S strain, it has a mucus coat, whereas the rough R strain, that is doesn't have a mucus coat. Now what Griffith did first, S strain was injected in a mice. When A strain was injected in a mice, the mice died. Next, R strain was injected in a mice, the mice remain alive. Next, what he did, the S strain was heat killed, and then again it was inserted into a mice, then the mice lived. Now what they did a combination of second and third one. So A strain heat killed one is added up with the R strain, that's a live one, and when it was injected in a mice, and they were shocked that the mice were dead. Okay, so you can see the first figure. Living S strain cells is found in a heart, whereas in the next no bacterial cells found in the heart. In the third, you can see no bacterial cells found in the heart, and the fourth figure you can see living S strains cells are found in the heart. When A strain hit kill and R strain live was injected into mice, the mice died. So he concluded that some transforming principle transferred from heat killed S strain to R strain. It enabled R strain to synthesize smooth polysaccharide coat and become virulent. This is due to the transfer of genetic material. The next experiment, there is a biochemical characterization of transforming principle is given. It was given by Oswald Avery, Colin MacLeod and McCarty. So that means Avery, MacLeod and McCarty gave the biochemical characterization of transforming principle. So when Avery, MacLeod and McCarty worked to determine the biochemical nature of transforming principle in the Griffith experiment, they purified biochemicals like proteins, DNA, RNA. Again I repeat, Avery, MacLeod and McCarty worked to determine the biochemical nature of transforming principle in Griffith. What they did, they purified the biochemicals, that are protein, DNA, RNA, etc., from heat killed S cells using suitable enzymes. So you can see one test tube where carbohydrate are removed. Okay, you can see in the first enzyme the protease was given, and we could see that no protein was there. The second experiment, RNAs were given, and we could see no RNA were there, because RNAs degrade the RNA, protease degrade the protein. And the third one, you can see DNAs were given where no DNAs were produced or seen. So they discovered that the digestion of protein and RNA, that is using protease and RNAs, did not affect transformation. So transforming substance was not protein or RNA, but the digestion of DNA with DNAs inhibited transformation. So this means that DNA caused transformation of R cells to S cells, that is DNA was the transforming principle. Okay, so when DNAs were added, they could see that no transformations were seen, so they told that DNA is only responsible. Next, the third experiment is Hershey-Chase experiment, that is a blender experiment. So what they did, Hershey and Chase grew some bacteriophage virus on a medium containing radioactive phosphorus, that is P32, and some other on medium containing radioactive sulfur, that is 35S. Okay, one is 32P phosphorus, another is 35S sulfur. So viruses grown in P32 got radioactive DNA because only DNA contains phosphorus. Viruses grown in S35 got radioactive protein because protein contains sulfur. So we know that the proteins contain sulfur, and whereas the DNAs contain phosphorus. So here you could see a medium containing bacteriophage having protein labeled with sulfur. And next you can see here in the blue color solution, you can see medium contain bacteriophage having DNA labeled with phosphorus. Okay, and you can see the bacteriophage there. So these preparations were used separately to infect E. coli. These two preparations were used separately to infect E. coli. So after infection, the E. coli cells were gently agitated in a blender to remove the virus particle from the bacteria. Then the culture was centrifuged to separate lighter virus particle from heavier bacterial cells. Okay, and the culture was centrifuged to separate lighter virus particle from the heavier bacterial cells, because we know that viruses are lighter and bacteria are heavier as compared to virus. So you can see first is the infection, then blending, and then centrifugation. So in a medium, the first one you can see medium containing bacteriophage have protein labeled with S. Next they are injected, blended, and you can see like bacterial pellets at the bottom and radioactivity like phase protein in the liquid. In the next one, in the blue color solution, you can see medium containing bacteriophage having DNA labeled with phosphorus, that's P32. Then it was injected and blending was done, and centrifugation, you could see that in the pellet the radioactivity, the phase DNA in the pellet. So bacteria infected with viruses having radioactive DNA were radioactive. Okay, that is DNA has passed from virus to bacteria, and bacteria infected with virus having radioactive proteins were not radioactive because protein did not enter the bacteria from the virus. Okay, so you can see the diagram. So this proves that DNA is a genetic material. So here you could see that no radioactivity like 35S sulfur was detected in the cell, and this side the radioactive 35S was present in the supernatant. Okay, so both the figures were given, you could see. In case of S35, no radioactivity detected in cells, and radioactivity is detected in supernatant, whereas in case of P32, radioactivity detected in cells and no radioactivity in supernatant. So this proves that DNA is only the genetic material. So you could see the steps like infection, blending, and centrifugation. First, a bacteriophage is taken, and infections were done. Okay, infection, blending, and centrifugation are the three processes. The first infection is done, then blending is done, then centrifugation. So after centrifugation, they could see that in 35S sulfur no radioactivity is detected in cells, whereas the radioactive supernatant, okay, was seen, whereas in case of radioactive phosphorus detected in cells and no reactivity in the supernatant. So we could see some properties of genetic material. So a genetic material must have the following properties. So first property is they should have the ability to generate its replica, that means replication should be there. And second property is they should be chemically and structurally stable, that means there should be a stability in chemical and structural. And third property is they should provide mutations needed for evolution. And fourth is they should ability to express as Mendelian characters. So these are the four properties of genetic material. First, they should have a replication power. They should be chemically and structurally stable. They should provide mutations needed for evolution, and they should have the ability to express as Mendelian character. So you could see under the properties of genetic material, two columns are given. One is reason for stability, and next column reason for mutability. So the reason for stability is there less reactivity, okay, of DNA, and reason for mutability is high reactivity of RNA. So this one is under reason of stability, they are double stranded, but this side is single stranded. So under reason of stability, they have a presence of thymine, and this side they have a presence of uracil. And under reason of stability, they have absence of 2' position OH in sugar. On this side, they have a presence of sugar in second position. So you can see the figure here. In the second position, under deoxyribose, there is a oxygen group is absent, whereas ribose group there is oxygen group present. So let's see the properties of genetic material. Like thus, RNA is unstable. So RNA virus, example, bacteriophage tuberculosis virus, mutate and evolve faster. DNA strands are complementary. On heating, they separate. In appropriate conditions, they come together. In Griffith experiment, some properties of DNAs of the heat-killed bacteria did not destroy. It indicates the stability of DNA. So you could see left hand side bacteriophage figure, and right hand side you could see the tobacco mosaic virus. So you could see a DNA is there. First it had given heat and it had unwinded, and the strands were separated and double helix were reformed. So for the storage of genetic information, DNA is better due to its stability, but for the transmission of genetic information, RNA is better. So you should know these things. For transmission of genetic information, RNA is better, but for storage of genetic information, DNA is better. So RNA can directly code for protein synthesis, hence can easily be expressed. So hence it can easily express the characters. DNA is dependent on RNA for protein synthesis. So DNA is always dependent on RNA for protein synthesis, because we know the central dogma, that is DNA is converted to RNA and RNA is converted to protein. So first is a transcription when DNA is converted to mRNA, and when mRNA is converted to protein, that process is called translation. So we could also see that when RNA is converted to DNA, that is also called reverse transcription. Takes place by presence of enzyme reverse transcription, transcriptase. Okay, so when RNA is converted to DNA, it takes place by presence of enzyme reverse transcriptase. Okay, so again I repeat, when RNA is converted to DNA, it uses an enzyme that is reverse transcriptase. So entire process of transcription and translation is called central dogma. Thank you, student. This was all about the entire lecture. You have discussed about the topic search for genetic material. This was from the chapter molecular basis of inheritance. So stay at home and watch the entire video lectures to gain basics. So again, we'll meet in the next class. Thank you, students.